terms of what we know about biology, we're probably 17 to 20 orders of magnitude away between what we know and what we don't know.
We want to see foundation models explode in biology that can understand not just one protein's function, but potentially any protein or any gene, any genome, in any context. We don't have that in biology yet. And so for us to get that right, we need scaled compute, scaled models and their parameters and architectures, and finally scaled data.
I think we are living in the most exciting time in human history because I don't know what's going to happen next. And I don't think anyone does know because the rate of change is fast-paced, really fast.
All right. Is this on? Hey, everyone. So excited to be here at South by Southwest. This is Decoding Nature, How AI is Learning to Program Biology. I am Sharon Goldman. I'm a reporter covering AI at Fortune. Really excited to be here for this really fascinating panel. I'm going to ask each of you guys to introduce yourself and what you do and what your work is, and then we'll dive right into this. Glenn?
Great. Well, hi, everyone. Is this working? Well, hi, everyone. Thanks so much for coming here. I did just realize as we were watching that I'm wearing the same shirt as I was in that film. I promise I have changed it and washed it since then. I'm one of the founders of Basecamp Research, and we're building a new generation of foundational AI models that, unlike your ChatGPTs, we're not understanding a human language, we're understanding a biological language. And so we collect enormous DNA datasets, we train these models on these DNA datasets, and then we apply that to generate new medicines. And I'm very, very lucky to call Alicia and César our collaborators that we've been working on through the last couple of years with. And very excited to have them join us.
I'm super excited to be here. So I'm Alicia Frame. I run the post-training product for Microsoft. So my job is to figure out for Microsoft internal companies and third-party companies how to make it simple and easy to customize large language models, whether they're open source or OpenAI models, for novel use cases. So retraining those models with your own data for your own scenarios, really unlocking the power of data sets to customize AI to different use cases. And I'm super excited to be joined here by practitioners like César.
Well, very happy to be here as well. I'm César de la Fuente. I'm a professor at University of Pennsylvania, where I run a research lab whose primary focus is to use the power of machines to accelerate discovery in biology and in medicine. And one of the things that we've done recently is we've developed AI models to mine the code of life to try to find new potential therapies. And so that's where our collaboration began with using the incredible data set that Basecamp has collected, which is completely unique, and try to mine it to see if we can find new antimicrobial molecules that we can use to counter some of the most dangerous infections that we're facing, right, as humanity. So wonderful to be here. Looking forward to the conversation.
Yeah, this is something that I love to talk about. I feel like this is fascinating work that we only have an hour, but I feel like I could listen to you guys talk all day. But over the past couple of years, AI for science is really considered one of the most optimistic use cases for AI. The idea that AI could discover new medicines or unlock entirely new biology. But up until this point, my understanding is that many scientists say that so far AI has really been about making research more efficient or faster, helping analyze data or speed up experiments, or something like DeepMind's AlphaFold, which is really about predictions. But the idea of fundamentally discovering new things has not quite gotten there yet. But Basecamp Research is really on that frontier, which is what we're here to talk about. So Glenn, I'd love for you to tell the story, which I find just so fascinating, about your team collecting DNA from ecosystems around the world and then training AI models on that data. Can you talk about what it took to do that, what you're trying to build, and how that then can speed up science to actually discover new biology?
Yeah, absolutely. And I think you're absolutely right. Most of the use cases in biology and life sciences right now for AI are fairly operational, saying how do I do the existing R&D process, but how do I make it more efficient and faster and better? And I think that's great. That's really important work. But I think that's a product of the type of models that we've had access to. If you think of your Geminis, your Claudes, your ChatGPTs, they're really powerful because they're trained on the internet. And so this corpus of human knowledge represented in text, image, and video. But what it means, though, is that they'll only be good at human tasks to some extent, or at least tasks that are represented at some level in that data set. When we move over into biology, if we want to go beyond just being able to optimize existing processes, we really have to focus on exposing these models to net new information and net new biology. And this was one of the early insights we had 5 years ago when we founded the company, that we so far as an industry or entire world, we actually know so little about biology and our natural world. And that really comes down to the limitations of existing data sets. So where ChatGPT and the like have had the internet, in biology, we literally don't have anything equivalent to the internet. Now, maybe just to put some numbers on it, they're estimated to be trillions and trillions of species on this planet, of which we've sequenced only a few 100,000. And by sequenced, I mean we read the genome and therefore we can understand what these organisms do. We've only done that for a few 100,000. If we run the maths on that, our CTO has a great analogy, which is if all of those species were represented as the Atlantic Ocean, we've only discovered 5 drops of water. And so that gives you a sense of that enormity of what's undiscovered out there. And so when we founded the company, we had an unusual start to some extent. We started the company on an ice cap where we were living up there it's called the Vatnajökull ice cap. It's where Interstellar was filmed in Iceland. And we're living up there for a month doing this DNA sequencing. We were trying to discover new organisms and new life forms. And the thing that was totally shocking to us as we were going through this expedition is the majority of the data that we generated was so new to science that with the best tools available, we couldn't say if this was an animal, a tree, a microbe, or a virus. We couldn't even assign a kingdom of life to the sequences that we were discovering. And that was firstly just incredibly exciting, but also spoke to a limitation of those public data sets and a need for us to go and discover vastly, vastly more about our natural world and about biology if we are to expose these models to it. So I'll just sort of finish up by saying where we are now, is we have collected this kind of data set. It's about an order of magnitude larger than public data DNA data sets today. And with the help of Alicia, we've trained this enormous model called Eden. It's very similar to the GPT-styled models, but trained only on DNA language. And then we worked with César to use this model to see if we could solve some really interesting problems in therapeutics. But I'll leave some of that as a teaser for later.
Amazing. Yes, we'll get back to that. But Alicia, you work on the infrastructure side of what makes AI for science possible. Everything is about getting it to be at scale, of course. So when you hear Glenn talk about this effort to take massive data sets and foundation models for biology to discover new things, can you explain to all of us more about what makes AI for science fundamentally different than what we think about when we use ChatGPT? Like, are you actually prompting these? Is it a language? Are we prompting it in the same way?
I mean, in a lot of ways, and I wince when I say this, but things like ChatGPT, Claude, they're really easy mode of AI, right? So if you think about what are they under the hood? ChatGPT, these large language models, they're language calculators. Given a word, what's the next most likely word? Words work sequentially. If you have enough words, you get a sentence. Enough sentences, you get a paragraph. And for better or worse, most of our language, whether it's text, whether it's videos, images, it's all digitized. So it's really easy to consume. It's publicly available. And as humans, we're really good at putting everything out there. We don't only write down the most interesting words or the best words. We have everything digital ready to consume at scale. And what really unlocked these large language models was just training on huge volumes of data. And the second piece to that is validating it. And it's really easy for you to look at something and be like, yeah, that's a word, that is not a word. Or that's a valid sentence, this is a valid paragraph. Or if it's a coding model, this compiles, this doesn't. So if you think about traditional large language models, they are easy mode. They're easy sequential building blocks, and it's really easy to tell, is this right, is this wrong? When you think about science, every single step of that process is a lot harder. So you can simplify it to, protein, function, cellular process. Sure, and you could say, well, I have one protein, what's the next protein? It's not that easy. Proteins operate, you have a sequence of amino acids, those fold into 3 dimensions. We've already gotten something that's much harder than words. And you already mentioned this, we have 5 drops of the ocean, but those are the 5 drops that were interesting to us. They are 5 drops of a shark. So in science, you publish what's interesting, what's groundbreaking, what's relevant to you. We haven't digitized everything else that's out there. And those data sets are private. They are owned by companies like Basecamp or Eli Lilly. A lot of information is siloed. So we're starting from a disadvantage in that the data doesn't exist, the data is sparse. It assembles into something much more complex than language. And then you think about validating it. It's easy to say, this is a word, this is not a word. But it's harder to say, this protein structure is valid for this task, or I have actually designed a cure for cancer. The validation loop is a lot longer, a lot more expensive, a lot more complex.
Okay, not everyone thinks it's that hard, I understand. Just a couple days ago, I believe there was a story that went viral about an Australian tech entrepreneur who used AI to help create a bespoke cancer vaccine for his dog, Rosie. So you're saying it's not that simple? That's a little hyped up?
Am I here to be the Debbie Downer on the panel?
Well, I'll ask César also, but what do you say, Alicia, first of all?
So I think that's like a great example of, AI can give you directionality, right? So you could say to ChatGPT, my dog has cancer. Can you think of a cure? And ChatGPT will probably be like, a novel cure? Maybe an mRNA vaccine. That is about as novel as saying, hey, ChatGPT, I have a headache, what should I do? And it's like, you should take Tylenol. It knows historical data. It can give you the right answer. But then you have to go into the lab. And the piece that was kind of underplayed in that story was, well, someone sequenced that dog's genome, and then someone found the mutation, and then someone synthesized the mRNA to give it to the dog. So this is a great example of AI kind of nudges you in the right direction, but then there's so much to do as a practitioner in the lab to actually make it real.
What do you say, César?
I agree. I mean, I think that what we see in the media a lot is that AI is this magical thing that is capable of emergent properties that were never anticipated by the human brain. And I think that's not the whole story. I think the whole story is that the best AI models are trained on really good, high-quality data, right? And I mean, I think Basecamp is a great example of that. They're scaling this high-quality data set in my own lab. Academically, we've done that as well. In the last decade plus, we've been creating our own high-quality data set to train models. And I think the successful equation is really good data plus AI systems equals success. And I think that maybe is not emphasized enough, surprisingly to me. But if you look in detail at all the success stories, or most of them, at least in science, they all have really good data sets behind them. So I would.
I wonder, César, if you could also, from the scientist's perspective, maybe connect for all of us this work that Basecamp is doing and that you're doing in your lab to some of the milestones around biology that people may remember, like around 20 years ago, the Human Genome Project that mapped the entire human genome, then CRISPR revolutionized gene editing. So what changes when researchers have access to these models that are trained on these huge, massive data sets for biology, like the ones that Basecamp is building? Like, what kinds of discoveries become possible that warrant? Like, is there an example from your own work?
Yeah, I mean, if you look at the recent history of science, we had molecular biology, right, where we learned how to manipulate one gene or one protein at a time. Then in the 2000s, we entered into the era of synthetic biology, where we learned how to do that in a sort of high throughput way, manipulate many genes, many proteins at a time. And now, because of our ability to create these incredible data sets and advances in compute and in algorithmic power, I think we have entered this era where we can do a lot of this on the computer. Some people call this digital biology. I mean, we're doing that in my own lab. I mean, Basecamp is doing that. We can do prediction, we can do generation tasks on the computer with high level of accuracy, at least in narrow fields. in my own work, we work on antibiotic discovery to try to come up with new molecules to target infections. And so we've now gotten really good at that narrow field, right? Things that these things unlock, some of the models like Eden, is that we can now understand evolution better, actually. Evolution is this incredible planetary scale optimization process, right? It's the longest optimization experiment that we have access to. So it's really amazing. And the fact that we have only sequenced a tiny fraction of every organism that has existed or that currently exists in the world is quite mind-blowing to me, right? Because that directly means that we don't fully understand evolution. We don't understand the world around us. We don't understand nature. So I think some of these data sets, what they might unlock is that we might finally be able to understand better how evolution did certain things, meaning how different molecules evolved or mutated over time, and perhaps shed light into why evolution did those things. And so in terms of the context or the environmental triggers that perhaps did that. And so I think it's an opportunity to learn more about that.
Glenn, like what? Like what kind of, like if I think about the kind of data that you picked up on your expeditions, what would help you understand evolution more, for example?
Yeah, well, I feel like evolutionary biology is having a resurgence now. It used to be the sort of domain of ecology or developmental biology or things. But I really think we should view evolution with a slightly different light. Everything that you see around you that's living is a product of that optimization process that you're talking about there. And what's beautiful about it is it's scaled, like I said before, these are trillions of species that are out there that we could be studying. And so when we need data sets of the scale of the internet, evolution is one of those axes you can push on. But it also follows a very clear code and language. And I think this is something that led to the fields of synthetic biology and now digital biology. But I think in this newer AI era is even more important that there is an underlying logic to evolution and DNA. And this is why we really focused on DNA as our main data modality to scale on, because of that language, that scalability, that ground truthing for when you find something in the natural world. Now, it's not to say that there aren't other data sets in biology that you need. And of course, over time, you will have to layer on, RNA data and image data and cell data and organ data and things like this. But I think we really think about it more like a data pyramid. And you want to start with the broadest, largest possible base, very similar to how GPT, it finds value in your random Reddit comments that seem completely disconnected, but it lets it learn a language. And then once you've learned a language, sure, then we can start layering it with more specific and smaller and more relevant data sets. I actually think there's a parallel there with how humans learn.
So I actually have a PhD in evolutionary biology, past life before AI. And I think that's like the perfect analogy. But one thing I want to kind of poke at is this notion of evolution for optimization. And so like thinking back, putting on my old academic hat, we talked about the Panglossian paradigm of like, why do I have a nose? Oh, it holds up my glasses. No, I have a nose because I need to breathe and a nose kind of works for that. And like one of the kind of fallacies in evolutionary biology is that you're always evolving to an optimum, the best state. But really there's random variation and you're surviving. Does this kill me? No, cool. This gene is going to be carried on. And when I talked about kind of data sets, I said, a problem with scientific data is that if we look at academic publications, clinical trials, we're only keeping the useful, interesting data. But evolution generates tons of variation and lots of trial and error, and it gives you that diversity that is your random thread on Reddit, or your Yahoo Answers, or your Tumblr accounts. And from that diversity, the models can kind of synthesize those higher order things that don't get killed. And I think evolution, kind of coming back to it, feeds into that just variability and the data sets you need to make sense of it. So it's more than just optimization.
Yeah, I was just going to add one little thing, that in research, typically we look down upon negative data. However, we now more than ever have realized that negative data is incredibly valuable, particularly if you want to train models that are good.
What do you mean by negative data in this?
Negative data, so for example, when we do screens in my lab to try to find molecules that might be potential medicines to target infections, the ones that don't work, we typically don't, we don't publish that. We don't publish the ones that don't work necessarily, or at least traditionally, that's how it has been done. But now the culture is shifting in research as well, because there has been a resurgence of, well, we actually care about negative data. In my own lab, when something doesn't work as intended, the PhD students might get a little bit depressed, but I look at them and I'm like, no, this is actually hugely important data that you just generated to train our models. So it's like a win-win for me in the sense that I can motivate people, but it's also actually true. And so then it works, right? And so in my lab, we value negative data almost as much as positive data in the sense that potential hits that we find in our projects.
I'm not sure if this makes sense, but in reporting on AI, for years, there's been so much pushback on the idea that, for example, ChatGPT and Claude basically scrape the internet and using all these Reddit posts. And I feel like this is kind of an example where you can really explain the benefit of having so much data. Can you talk a little bit about the rep of data gathering and why I feel like it's important for people to understand why it's so important to gather this scale of data in biology.
Well, I guess linked to that, something we think a lot about is the process of gathering that kind of data. I think in the natural language world with the internet, you have a slightly weird paradigm now where most of the data does exist. You just have to scrape it or, in some cases, steal it, and that's not good. But you have to create agreements to go and access this data, and it's very complicated and mixed matched because the internet wasn't created for this. It was created for different reasons. We have a nice opportunity in biology to some extent because those data sets don't exist. We get to build it from scratch, and which means we can get some of these properties like data ownership and sovereignty around data and traceability around data. We can just fix that to be right from day one. And so what that means for us is all of this data comes from the world. For us at Basecamp, it comes from 31 countries around the world, countries like Cameroon and Malawi and Costa Rica and Antarctica as well. And we actually create access and benefit sharing partnerships with every country we go to. We don't even set foot in the country unless we have full negotiated rights to this data. And to get those rights, we do that in exchange for value. We say, near term, we will work with you to establish labs and capabilities and training, and we'll share data back with the country and the researchers. And in the longer term, there'll be a royalty stream as and when money gets made from any uses of this data that come downstream from that. In fact, we've actually passed back over 100 royalties to places around the world. So for us, I think it sort of we can set a new precedent and we can be better than the sort of AI world has been to date because we get to build it from scratch. And I think that's just a really exciting sort of route to scale even more data because all the incentives become much more aligned.
Got it. That sort of leads me to another kind of ethical question. I was curious because when I think of CRISPR, this is a project that has long had a lot of ethical concerns, what this data will be used for. So if AI can design biology, doesn't that raise concerns about safety and misuse as these models become more powerful? So how do we make sure that they are used responsibly? I was wondering if each of you have any thought on that.
So I can talk a little bit about it kind of from the Microsoft perspective of we have a large investment in responsible AI. And our focus is on, when we offer large language models for enterprises, they should be safe, secure, and private. And so kind of standard practice for a lot of these frontier labs, whether it's OpenAI, Anthropic, is that models are aligned before they're sent out to the public. So they are trained on everything on the internet, but they've been aligned to respond helpfully, to not respond to harmful content, to not agree to generate bioweapons or hate speech. And that kind of goes into that process of the internet is full of all kinds of things that we don't want to perpetuate in these models. So a lot of work goes into these safety benchmarks for a foundation model in terms of what it'll do, what it will provide, and how it's kind of oriented to respond. And then when we talk about custom models, which is kind of what my team focuses on, so you take an off-the-shelf model, you train it, we work really hard on automatic detection of things like jailbreaks. So if you pick up a model, there's lots of red teaming papers out there that show, here's how I can break all the safety alignment for model X. We have a pretty rigorous benchmark of tests that we run before a model can be deployed on Azure to make sure that it hasn't been jailbroken to do hate speech. And we have content monitoring to see if people are using these models to develop bioweapons, hate speech, all kinds of harmful content. And so that's pretty standard in the industry. I'd love to understand more about, from the biotech side, do you do similar things with Eden?
Yeah, it's a really interesting question. I think the difficulty comes to one of the first things you were saying on the validation, and we don't speak DNA, we don't speak biology natively as humans, and so it can be hard to work out, is a model doing something I don't want it to do? Is it coming up with a virus that I don't want it to be coming up with? That detection's kind of really hard. So we make sure that we're being as transparent and open as we can with any regulators in the space. We see our role in the industry as saying, let's push the forefront of what these models can do and be very transparent with the results with the right regulators and entities that need to think about this. Because I think there's a lot of dual-use technologies, and AI certainly is one of those things. But we also, the upsides and the downsides sort of need to be taken into account with each other. But biology, it's a hard one.
César, is it something that you think about in academia?
Yeah, in academia, we've thought about it in the context of some of our projects. So in one of our projects, we develop AI models to explore, for example, extinct biology and to find functional molecules encoded in extinct genomes and proteomes. And we found, for example, new antimicrobials in the proteome of the woolly mammoth or Neanderthals.
I was just thinking about a woolly mammoth, and you just said it. Okay.
So we've sampled all of ancient biology, and then we found molecules that are unique to ancient biology. That when we try to align them with molecules that exist in the world today, we don't see overlap. And then we've synthesized some of those in the lab to try to cure infections. But then we started engaging with bioethicists and philosophers of science to see if that's okay for us to do that, like resurrecting molecules with chemistry in the lab to try to fight infections that are killing humans today. Is that a valid, ethical reason? And I've become really interested, and we've had many discussions with bioethicists, and I think it's important. It's impossible, like what Glen was saying, it's impossible for us, doing the research to anticipate every potential problem. But I think having conversations at large, not only in academia, like industry, government, and so on, but also with the public at large to try to understand, what are the limits, where are the boundaries that we feel comfortable with, and how much are we comfortable with pushing in one direction or another. And in the last couple of years, I've signed a number of petitions calling for the safe use of AI in biology. And one of the specific things that we've incorporated in my lab is that if we find molecules that are predicted to be biotoxins or something like that, we don't proceed with those projects. So that's one specific intervention that we've incorporated. But there are probably many others that I can't even think of. And so we're very much aware of our limitations and our blind spots. And so I think that's why I welcome conversations like this, hopefully in the Q&A session and just interactions like this to try to figure out what we feel comfortable with.
I guess it's also important to remember that there are regulatory frameworks. We can't just type into a computer, give me a cure for cancer, and then take it overnight. There are clinical trials, there's safety validations, toxicologists exist. So there is some guidance out there.
I think it's a circular argument, but I think AI can help with this to some extent. One of the models that César's lab has built is called Apex and enables you to take a sequence of a peptide and predict its function in the real world. So we can use that mostly to say, what is the good function? Will it kill a bacteria that is infecting a human and we don't want it to, a critically difficult bacteria? But we can also probably flip those models around and say, is it creating something that we don't want it to create? Is it creating a toxin? Is it creating something that could cause harm to a human? And I think those AI models, you can sort of responsibly use them in both directions to up-select the ones you care about and make sure that they're sort of down-selecting the ones that you don't care about.
Well, Glenn, you've talked about this as programming biology. In the case of the Eden model, I understand it's programming gene insertion. So I think we have a little video here, but I'm kind of hoping we can share what that idea means in practice, like what we're really seeing. So I think that we're going to pull it up here. Can you see it? Yeah.
Yeah, I guess just maybe to intro this, I suppose, for us, programming biology is really just taking a step away from what the current drug discovery process looks like. And I even think that phrase drug discovery speaks to its limitations, that we often are doing a lot of trial and error at the molecule design phase. We're stumbling across molecules. Like even today, 60% of molecules in the clinic are just happen chance discovered from nature. And we wonder how they work. We do some early lab tests to see if they work, and if they work in the lab, fantastic. We'll maybe inject them into a very, very small number of patients at very low doses to see if they don't die. And if they don't die, fantastic. We'll give them to a few more people, and if they don't die, we'll keep going up that scale until we're comfortable we've given them to enough people who've not died. That just speaks to our fundamental lack of understanding of biology. And we either believe that that is the best we can ever do, or we believe that we can get to a point where we do understand biology enough to predict some of those outcomes. Now, if we can predict those outcomes, that's where the programmability starts to kick in, where you can say, I can intentionally design a medicine based on a disease input where I feel I have predictability on how that will behave through that process. And even today, there are some pharmaceutical companies, I think Novo Nordisk has gone on record saying, we eventually, in a very short period of time, a matter of years, want to skip animal trials. We think we can have enough predictability of how a molecule will behave that we don't want to be putting them into animals anymore. We want to skip that step. So programming biology is all about understanding that disease input, and with a programmatic sort of mentality, design a medicine and have predictability over that. So yeah, we're happy to play this, and we can sort of talk through this just to give it some life. Yeah, so this is an example that we did at Basecamp with Eden. We could type in a disease, genetic disease. In this case, we're just showing hemophilia B. And what the model's doing is it's looking through the genome of a patient that had that hemophilia B to identify the gene that would be responsible for that. So that's that disease biology input. The model then starts reasoning through different proteins it could design to go and treat that disease. And in this case, it's landed on this white and orange protein called a recombinase. And then what it's doing is it's bringing in a healthy copy of that gene and doing a simple recombination. It's just inserting that healthy copy of the gene where that diseased copy used to be. And in that way, you end up with a correct genome. So this is a fairly simplistic example, but it really speaks to this idea of programming biology for genetic diseases, where we have 10,000 genetic diseases today where we know the cause of them. We know the disease gene. We just don't have a way of programming a new medicine specifically to go and treat that. And I think, if we really sort of play this out into the future, this is really what we mean by personalized medicines in the long term. This is something that's been talked about a lot for decades, that we could get to this idea of programmable personalized medicine. But the other side of personalized is it needs to be programmable. We need to be able to understand the person, understand the disease, and in response to that information, generate a medicine. And that's something that, again, just in this one example, but something that we're really, really pushing on with these new models.
Is this idea, would this make, for example, curing very rare genetic diseases that it hasn't been economically viable to work on a cure? Is that a big part of this potentially?
Yeah, certainly. I think the phrase it's uneconomical to treat a disease is just a function of the cost it takes to treat that disease, and most of that cost comes from those clinical trials. So again, if we ever got to a point where we really did understand the biology and had predictability over it, and eventually you could have such high levels of predictability, you could skip levels of clinical trials. I'm not at all claiming we're here yet as an industry, but that is a future that could exist. In that case, a lot of diseases become economical in that sense. It costs you far less to develop a cure for a small number of patients who have a very rare disease. You don't have to go for only blockbuster drugs at only huge patient populations. In fact, there was an example of this recently. Perhaps people in the audience have come across it. It was a child's. It's been named Baby KJ, which is sort of the initials of the child, that was born, I think it was in Pennsylvania.
It was at Penn, at University of Pennsylvania in CHOP, yeah. Oh.
Absolutely. I mean, maybe you can speak more to it, but baby was born with a very rare and individual genetic condition that meant life expectancy would be rather short, and in a matter of 9 months, a personalized gene editing therapeutic was built and made and designed for that child. It was approved by the FDA in that window of time, and it was administered to that child. And I think, and as far as I'm aware, unless information has changed, that child is healthy as they are today, and is even up on stage in some conferences, not shouting the baby, but the parents of the baby sort of shouting about the successes that's happened there. That's one example, but I think it speaks to a real movement that could happen now for rare diseases.
And César, your work is not in gene insertion, but your example around the antimicrobial work that you do and other things. Can you talk a little bit about how that is another example of programming biology?
Yeah, sure. So we're facing this big problem now where we have bacterial infections, right, that are associated with 5 million deaths every year around the globe. That number is projected to double by 2050 unless we come up with new antimicrobial molecules, and is projected to become the #1 cause of death in our society. So huge problem, critical problem around the world, and a huge need to come up with new molecules that we might be able to use to combat antimicrobial resistance or these drug-resistant infections. So the work that we've done with Basecamp is we've applied some of our AI models to go into their incredible data set to try to find new molecules that evolution has created through the evolutionary process that might be helpful in our fight against some of these infections. And indeed, we have found a number of them that work really well in the lab, and we're currently doing preclinical work to see if they work in mouse models of infection too. So we'll probably have in a couple of weeks, we'll be able to report on that probably. But it's just another example of if you scale how much biology you sample, also it's something we've seen in our own research. You get a higher yield of function of molecules that do what you want them to do. And so that also ties in really well with the thesis of Basecamp, that if you really create this incredible data set, I do believe that you'll be able to find molecules that can do a number of things, including for indications in medicine and biotech and many other areas, right? Carbon capture, you might be able to find things that do carbon capture better than we can do it today, or a number of different semiconductor materials, you might be able to find them there, room temperature semiconductor materials. We're working on some of these areas in collaborations, and so why not? I think the potential is huge. Yeah.
Why not indeed? But Alicia, I want to ask you about your work at your role at Microsoft and your work at Basecamp. We often hear that scalable AI, it's not just about the data, it's about the compute, it's about the infrastructure, kind of, I don't know, maybe the less sexy side of this conversation. But, NVIDIA providing GPUs and Azure providing that orchestration layer, and then Basecamp providing the data sets. Can you talk about what it takes to bring that all together at scale?
So I'm going to object to this being the unsexy part. Just depends on what your interests are.
That's right.
Infrastructure is really exciting, but I think this goes back to this notion, like if you think about, the Human Genome Project, right? That took hundreds of collaborators, thousands of collaborators across research, academia, industry to build something big. LLMs take lots of collaborators working together. If you think about like, how did we get to ChatGPT, right? There were research papers coming out of DARPA. There was Google doing research. There were academics doing research, and tremendous amounts of compute. And so none of these things are like, I can go to my basement and build it myself, no matter how good I am at vibe coding. What you need is you need a lot of compute. You need GPUs to run the workloads. You need an orchestration layer, which is very sexy, on top of those GPUs to execute your training jobs, make use of them, schedule them, restart them when they die. You need the infrastructure to store, retrieve, parse that data set. And you need collaborators who can validate the thing you've built and said, yeah, this is real, this is not. And I think when we talk about science for AI, it's really going to come down to the partnerships, right? If we talk about Basecamp, you've got NVIDIA with the compute, you have Microsoft and Azure with the infrastructure, you have Basecamp with the data. But it's not just that. You see collaborations like the Genesis Project or ARPA-H, bringing together a lot of the big players in industry and academia. And there's this sense that because so much of the scientific data is siloed, and it's still hard to get GPUs, and there are only so many people who are Kubernetes experts who can schedule these jobs. We all really do have to come together and collaborate on the entire life cycle, from compute to actual outcomes in the lab.
Awesome. Yes, very sexy. Sorry, orchestration. So we are going to take some questions at the end here. So just saying this now to get yourself thinking about any questions you'd like to ask these amazing panelists. But meanwhile, I'd like to ask Glen and César kind of what's the next step here. Glen, do we need more data? How much more do we need? What would be enough? How do you gather it? What's the next step here in this journey?
Well, I think what's been a really interesting journey in the wider AI space has been the establishment of scaling laws back in 2020, and really the industry leaning very hard into those. And what does it take to increase the intelligence of these sorts of models? And what are the big levers that we can pull? And early on, at least, it was fairly simple. It was really saying we need larger models, we need larger data sets, we need more compute. And if we just keep scaling, and I think OpenAI showed that over 7 orders of magnitude, these models keep getting better. It's more complex now, of course, but I think we're in that phase in biology now. And I know our lab and César's lab do a lot of work on establishing these scaling laws, which are unique in biology. They're not exactly the same as they are for natural language, but they do still exist. And one of the things that our Eden model was showing is we could change the scale of model, we could expose it to larger data sets, and we could work out what those parameters were that meant these models would keep getting better, if you like. And so while we're at 10 billion genes right now, we're making sure that we put plans in place to keep scaling that. This is not the end state. This is really the beginning of saying, we've got to this large amount of data, which has allowed us to establish what these relationships and scaling laws look like. Now's the time to put the foot on the gas and say, how much more can these models learn? How much more data can we expose them to? And where do these laws start breaking down? Is it 2 orders of magnitude more than where we are today? Is it 3? Is it 5? There will be probably some limit to this, as the field is seeing now in the wider AI space. But we are nowhere close to that limit right now, that's for sure.
You have to go on more expeditions?
Yeah, mostly to the Caribbean, I think, is where they...
Or Texas.
Or Texas, of course, yeah, Austin here. No, absolutely. I think this is the beautiful thing about evolution, though, is it really does exist in all the corners of the world and all the niche areas. And as part of this, we can bring in these collaborators from all these countries around the world and create these data assets and really sort of understand biodiversity at a much larger scale. And that's really the offshoot benefit of this. So yes, we're feeding it to AI models, but at the same time, we're just generating and sharing back with these researchers all of this new data about the biodiversity that is largely invisible if it's in microbes and in their soil, but yet governs so much of their ecology and things like this. So there's lots of offshoots to scaling data, not least of which scaling the intelligence of these models. So we're very, very excited to keep pushing on this.
How do you see that from the academic standpoint in terms of what you need?
Yeah, I would add that there is all this biological intelligence waiting to be discovered in the world around us that, by reading all these genomes, we'll be able to, I think, uncover and unlock a lot of that biological intelligence, which is different from artificial intelligence. We don't even understand how biology does things, and so I think that's really interesting. In our own work, the way we've done this is we've generated data sets in-house, experimental data sets to train specialized models to predict for antibiotic function, to predict for toxicity, to predict for stability, PK/PD profiles, all these things, right? And we're really good at predicting antimicrobial function. We're at 95+% accuracy in hit rate, which is an experimental metric that we've developed. So that tells us that if we train our models with more high-quality experimental data, our models can get really good at one point. So I think we've demonstrated that hypothesis. So now we're expanding that to all these other things that I mentioned, right? Like toxicity, all these other things that we care about when we're trying to come up with a new drug. And in parallel, we're also developing AI agents that can tap into each of these different specialized models. And I think in the future, we're going to see a lot more of that, where we have agents tapping into each of the models that we've developed over time that are incredibly good, like superhuman good at each of those narrow tasks, but then combining them all in a coherent way that allows you to do something really hard, right? Like creating a new drug, for example. That's a non-trivial endeavor. And so that's what we're trying to do at the moment.
What kind of timeline do you feel like this is on? Do you see it as kind of Would you describe it as a moonshot, or do you see it as a very practical, like we could really get here in a few years?
I think it depends on the endpoint that we're sort of kind of agreeing on here. But I think with César's example there of having, there are today lots of specialized models that are very good at individual steps. We have models that can sift through genomes and find interesting new targets. And very, very separate to that, we have models that can design new gene insertion or new antimicrobial peptides or other antibodies. Therefore, the orchestration to put those two things together doesn't feel wildly complicated, yet the result will feel wild. It will be effectively a single agentic system that can reason through the disease biology and generate interventions in the same breath. I think that's going to feel like it's a huge step forward, but I actually think technically it's not as far away as it will feel when it's here. I think it's very achievable.
Is there any real bottleneck that concerns you, like any big challenges that, if you're thinking even just over the next 6 to 12 months, is there anything that you just feel like, we need to get through this bottleneck to get where we're going? Is it the data? Is it?
Yeah, it's an interesting question. We from our perspective, I think, yes, the data is still a big limitation. I think we only know the tasks we can achieve based on the data sets that exist today. I think you have a lot of this kind of capability overhang as models get much bigger to say, we don't know what they'll be able to achieve as they grow and as we scale. That's both very exciting, but it is also a challenge. I think almost the value here for any scientist is to say, what is the quality of the question that you can ask these models? And that will just only get more and more important, and the creativity of that question that you can ask these models. I don't necessarily think, and maybe Alicia might disagree, but I don't necessarily think the technical route to building those models is such a bottleneck. I think we know what that roughly looks like, largely because the general AI field is a lot further ahead than in biology, so we've got a lot we can just learn from that. But I think, the implementation, the quality of the question, the sort of validation that you put around it, I think these are all still bottlenecks today that we can optimize on.
You've been describing like a kind of classical architectural question that we've been working through kind of in the general purpose LLM world for the last couple of years of, when you have a general purpose LLM, you have these emergent properties where I didn't train it to do that. I trained this to be a next word calculator. And remember 3 years ago, everyone was like, I had it write me a shanty about my middle name and pirates. And wow, isn't that exciting? But you got kind of all of these emergent, exciting properties from just training with enough data and having kind of novel things appear because the model in the embedded space was able to kind of draw parallels that would take many, many, many humans thinking, collaborating, working through things much longer to do. But then you're talking about these smaller specialized models and like agents and what's the trade-off there? And we talk about, these mixture of experts models, which are, we talk about reasoning models, GPT-5, the Sonnet series models, where it's no longer just one really big model. It's a mixture of experts where you have a chat model, a reasoning model, an SLM that can do classifier responses really fast. And kind of in the general purpose AI world, like we've seen a rise of those mixed models where for tasks that you know how to do, you kind of have a router. And working in the agentic space, we're actually seeing, I have customers with hundreds of specialized small models where they have high confidence that it'll do this one task really well. But at the same time, you have your general purpose model for the catch-all of someone has a new idea or we want something general. So you can kind of balance those trade-offs. And I think those emergent properties are what makes it really hard to say, where are we going to be in 6 months and 12 months?
César, is there any kind of holy grail opportunity that you see out there for your work that you're really hoping that this work with Basecamp will unlock for you?
Yeah, I think certainly identifying functional molecules at that scale, we can start thinking about, not only antimicrobials, but anti-cancer agents, things that modulate the immune system, drug delivery molecules, anything like that. So, it's exciting to think with that level of ambition, and I think I think future collaborations probably we might explore some of these things. Who knows?
Glen, do you see something like curing cancer? Is that a holy grail opportunity or goal for Basecamp?
Yeah, absolutely. I mean, it's a holy grail for the entire world and field. I mean, cancer rates are increasing right now, not least because I suppose lifespan is increasing, and that just creates even more urgency. What we're very excited about there, back to the sort of programming biology message, is almost all cancers are different to each other, and I think that's something that we underappreciate. We know that when we do diagnoses, but then when we come to treatments, we use effectively nuclear bombs of therapeutics like chemotherapies to still treat these. So sometimes when we talk about personalized therapies, we think of rare diseases, but really personalized therapies, I think, also absolutely applies to cancers, which are incredibly heterogeneous. This is where I'm very excited for AI to have a very, very large impact. It can both really understand the nuance of the disease and take in huge data inputs. It could take in potentially, whole lifestyle data from wearables and, huge data corpuses that would be more than any one scientist or team of scientists could sift through, but AI models can really make sense of that, and then pair that with models that can generate medicines and interventions in response to that data. This really is the holy grail for medicine, not least cancer, of course.
Well, I'd love to take a couple questions. We have a mic over there. Let's make sure, is the mic on?
Hello, is it
Yeah.
recorded?
And just say your name and who you are.
Hey, my name is Matthias. I have a question. So, I work in data and AI, but I do a lot of things. I write, I create, a lot of different things, and I'm really interested in this because I think it's the future. Well, it's the present too, right? So,
What is your question?
I'm about to ask it. So, what are the building blocks? So what are the tokens that you guys are dealing with? Like in language, we have, just their words. But here, I feel so many levels of abstraction.
Yeah.
You can talk about cells, DNA, RNA, and it builds all the way up to a life form. I'm thinking about interplanetary life design, creating a life form that can operate on another planet, or applications to permaculture and designing systems. So just the tokens.
Yeah, of course. If we want to be really reductionist, I think we can view almost everything that is emergent in biology as a product of the DNA that encodes that life form and the physical environment that exists around that, that governs how that genome might behave. That can be both cells that evolve near it, or the environment that it's evolving in, or whatever that might be. And so for us, the token that we use is an individual nucleotide of that DNA sequence. And this forms a code. If you sequence all 3 billion plus nucleotides of a genome, you're reading a language. It's just a language that we as humans don't speak. I can't converse to you in DNA, but it is a language, just like any other language. It has a left to right reading pattern to some extent. It gets more complex than that, of course, but that's where doing that kind of tokenization of DNA at scale, you can start to unpick some of that complexity. Yeah.
Thank you.
Hi.
Hello, Justin Kiggins, recently left the Chan Zuckerberg Initiative. Super excited about this work. One point that y'all touched on earlier is the bottleneck of actually validating this work in the lab. That ends up being laborious, costly work. I'm wondering if you could speak a little bit to the opportunities for LLMs, agentic approaches in helping to lower that bottleneck, either through improving individual scientists' productivity at the bench, lab automation, and those kinds of opportunities, and whether you're engaging in that kind of work yourselves.
So I think one thing that is important with LLMs is getting you the right hypotheses and the right data. So if you can kind of tip the scales on, I'm going to run an assay, and even we spent the last couple decades developing high throughput assays, in vitro assays that are faster than trying to give drugs to people and seeing if they die, giving you earlier signals. But if you can stack the deck so that you have more confidence in that hypothesis and that molecule and that protein before you run the test, then you can start to spend your resources wisely. But it's kind of a chicken and egg thing where you need data and you need test data to start to understand better. And I think that's probably where labs like yours come in.
Yeah, I think, at the beginning, you need to do the hard work. Automation can help, right? So you don't have to do as much human labor. You don't put as much into it. Once you train models that are specialized models that are 95% accurate for your particular objective function, then you don't have to do as many experiments. So that's what we're seeing for antibiotic prediction at least, and now we're moving that to other areas. But I think if you're talking about compressing timescales, I think automating, using automation for pipetting and things like that is helpful. And then trying to parallelize as much as possible the validation steps that you have. And then training really good models, so then you don't have to do experiments, or you minimize the number of experiments that you have to do.
Awesome, thank you very much.
Thanks.
Hello, thank you, good afternoon. My name is Ivan Oliveira. I come from Brazil, and I have two quick questions. One is regarding Brazil. When you talk about biodiversity, we're talking about 20 to 25% of all the biodiversity in the world. And also, we are the most genetically mixed people in the world. So I wonder, what's the role of Brazil in your research now and in the future? That's the first question. And the second is, it's great when we find the solution for whatever, cancer, autoimmune diseases. But what we see in the pharmaceutical industry recently is more personalized and every year less people being helped with the solutions. So finding the solution is not enough. Do you anticipate any kind of conflict to the big pharmaceutical companies that make a ton of money out of being so specific?
I could definitely start with the first question, that we would love to work with Brazil. We do not currently work with Brazil. We work with 2 South American countries and 1 in Central America. Brazil, absolutely, like you say, is the home of a huge portion of the world's biodiversity. And that number is probably underrepresenting all the invisible biodiversity we can't see in the soil and things like this. So yeah, let's maybe exchange details afterwards. It'd be great to meet.
I can maybe talk a little bit about the commercialization questions. I work with Microsoft Research. They have custom models. And one thing that we spend a lot of time thinking about is, like, how do you commercialize AI for science? If you look at a drug company, as you said, they're incentivized to find a molecule that they can push through clinical trials and make money off of to pay back that debt for coming up with the molecule in the first place, running the trials. And in some ways, it's a broken pipeline. We're finding smaller and smaller populations that can be treated for more and more money. I think AI kind of flips things quite a bit. You reduce the barrier to come up with hypotheses. You're able to rapidly think about personalization. And no pharmaceutical company is going to take a trial for a patient population of 1. But you start to think about, well, maybe instead I'm going to patent the mode of delivery, or I'm going to patent the technique to find that unique combination. And so one of those, forecasts, maybe not the next 6 months, is really seeing kind of different go-to-markets for these personalized molecules, treatments, diseases, and looking at the economics of discovery really differently, where maybe it's not the billion-dollar molecule, it might be the million-dollar hypothesis. And letting many, many different ideas bloom in parallel.
Somewhat vertically challenged. First of all, Alicia, I would like to say that orchestration layers are objectively hot. And so this is really hard to talk like, all right. As a programmer, one of the things that we're seeing in pretty recent times is that these models are becoming much more effective because they can self-validate, right? Which we've touched on this a little bit. So I'm curious if you see in the relatively new future, like Playwright for proteins, does it have a way to inspect what it's built and kind of validate? And maybe a little bit, should it? Like words can't express how undangerous and ugly web pages a protein that we're letting it cook up maybe is not advisable. But is there that way to improve that validation layer that you think might be kind of where the industry goes?
Well, I think, Glenn, you kind of got into that when we were talking about AI safety, where I said, well, for regular, degular LLMs, we run content safety filters and make sure you're not asking them to make hate speech. And you said, well, we can use models to check if these proteins have, genotoxic effects, if they're, potentially bioweapons. And a lot of this is going to come back down to how confident we are in these models that we're using as the checkers. So regular AI, LLM as a judge is a super standard approach to iterating. What's the quality of my result? I have another LLM review it and be like, or if it's code, I'm going to run my code in a loop and catch the bugs and improve it until it hits my target. I think to some extent we're going to see that with biotech. But the other thing that I think is going to be really powerful that I didn't get to talk about is reinforcement learning. That is the new hotness in AI of I don't just have, next word prediction. I'm not training a model to copy. I am setting the model at a task. I'm getting feedback, and then I'm adjusting the model. And I think that's where, we can do more with a lot less data. So if you think about a training data set for regular post-training alignment, that's millions of records. A reinforcement learning data set might be in the thousands because, the kind of gym style, make an answer, check if it's right, iterate, becomes so much more useful. And I think we're gonna see more interplay with wet labs and bench science where we kind of do these RL-style trials to build the confidence that the models themselves can judge their correctness.
Yeah, we are doing that actually, and it's working really well. Yeah. So RL, yeah.
Awesome. Let's take one last question. Thanks for waiting.
Thanks for lowering the microphone. My name is Angela. I'm from Austin. Quick question. It's more on the ethical side of things, and apologies if you already addressed this because I got here late. But I'm curious. I come from a financial services software, so people are just concerned about making money, and it's very black and white. So with what you guys do, how do you prioritize the projects that you work on when you don't just have to say, oh, which is the most profitable? Curious to hear.
Yeah, it's definitely an interesting question. And in some ways, working in therapeutics and medicine, it's a fairly easy task because clearly solving a disease and treating a patient is objectively a good thing to go and do. We do, of course, have to balance that with, what's the scale of the need and how broad can we go. But this is where we're excited about AI models because it shouldn't necessarily have to just put a cap on the number of diseases or the threshold of economic viability that you have to hit to be able to treat a disease. If we can have models reasoning across multiple different modalities, multiple different diseases at scale and generating really high quality molecules that we have high predictability over, then we can be a lot more permissive with the number of diseases and the rarity of those diseases that we can sort of have an impact in. So this is something I'm really, really sort of excited about. At the moment, I will just say, we haven't made our Eden models publicly available and, anyone can use them, and that's really for a safety effect. We don't really think these models yet are at a point where we can guarantee people can use them in only the right ways and not in any of the wrong ways. And largely that's because the outputs are things that are hard to validate in languages that we don't understand as humans. So we use the model ourselves. So we obviously pick the projects that we work with in the companies that we partner with and the academics that we work with. And yeah, we try to work on the hardest diseases with the most interesting molecules.
On that note, thank you so much. Let's give these guys a hand here. Thank you to Glen, Alicia, and César. And I'm sure they're available for any other questions that you have. So thank you all for coming.
Thank you.